Well, we're getting ready to enter a phase in medical neuroscience where we're going to be talking about the senses of sight, sound, and smell. And there's no experience that brings all that together than sitting around a campfire, holding a brain. So I look forward to these next few sessions with you, and if you have an opportunity to be near a campfire over these next couple of days to weeks, you might want to have the experience I'm having right now, where the sense of heat and warmth and the sound of the crackle and the dance of the flame and the smell of the burning wood all brings to mind these incredible feelings of warmth and relaxation and comfort. Quite an amazing bit of neuroscience behind all of that. So look forward to exploring some of these topics with you, and see you next time around the campfire. Welcome again to my home. It's a beautiful day here in central North Carolina, and I'm pleased to begin a series of sessions with you talking to you about the visual system. And our focus today is on the eye. What I have to share with you today pertains to several of our core concepts in the field of neuroscience. We again are confronted with the complexity of the brain as the body's most complex organ. And we again are going to be looking at the structure and function of circuitry that's largely determined by our genes. And these circuits will be foundational for the operations of the visual system in the human brain. And I can hardly think of any topics in the whole domain of sensory neuroscience that has fascinated more human beings over the course of our history than the sense of vision. And so I think when we come to this topic, we certainly are in a position to appreciate how our brain itself gives us this amazing capacity to wonder how vision works. Our learning objectives today begin with the factors and the neural mechanisms that account for focusing an image on the retina. So I want you to be able to describe them. I want you to be able to identify the 5 basic neuronal types that are found in the retina, and I want you to be able to state the roles of each in neural processing. I want you to be able to talk about sensory transduction in the visual system. So I want you to be able to characterize the molecular processes that underlie what we call phototransduction. And lastly, I want you to be able to discuss the responses of retinal ganglion cells to the onset and the offset of light, and the relevance of these receptive fields for the detection of light and shadow. Well, let's begin by looking at the anatomy of the eye. Well, I would just direct your attention to some basic anatomy here. So the eye is comprised of several layers and 2 chambers and some structures that are important for forming the sharply focused image on the back of the retina that are found within the eye itself. So in the anterior side of the eye, we have the cornea. The cornea is a structure that is responsible for actually most of the refraction of light that happens as light passes into the eye. So roughly 80% of the refractive power of the eye comes from the cornea. The remaining 20% comes as light passes through these middle structures near the anterior third of the eye, including an optical lens. And this lens can change shape through a process that we call accommodation. We'll say more about that in just a moment. Well, the space defined by this anterior portion of the eye is called the anterior chamber. And this anterior chamber is filled with a fluid called aqueous humor that aqueous humor is produced by some cells that sit right about in this region here. And that fluid then leaves what we call this posterior chamber between this ciliary muscle and the lens itself. And that fluid then fills up this anterior chamber, and it helps to support the curvature of the cornea. Now that fluid is typically drained by a trabecular network found in this lateral margin of this anterior chamber. Now, should there be a problem with the absorption of this fluid from the anterior chamber, then that can build up pressure within the eye, and that pressure has a way of damaging the peripheral parts of the retina. And as a consequence, in that eye, one might tend to lose peripheral vision. Well, let me try to illustrate the consequences of this, if I may, using a photo from my photo album. This was a memorable trip my family took many years ago. We're in one of the most beautiful sites in all of North America. This is Lake Louise in Alberta, Canada. And if a person viewing my family in this scene had glaucoma, sadly they would not be able to truly appreciate the full majesty of this scene, because their visual field might very well be reduced to just the center of their gaze. Well, let's now turn our attention to the process of focusing the image on the back of the retina. And this is a process that we call accommodation. So accommodation requires the active adjustment of the shape of the lens that sits in the anterior portion of the eye. So here what we see is an illustration of the lens in one of two conditions. In the unaccommodated vision, the lens is relatively flat. So this would be appropriate for far vision. And then over to the right, in the accommodated configuration, notice how the lens is considerably fatter and more round. So this will increase the refractive power of the lens, and this would be appropriate for near vision. Well, how does this work? Well, this is where the active elements of this ciliary body come into play. There is a muscle, the ciliary muscle, that surrounds the lens. And this muscle is attached to the lens through a series of fibrils called zonule fibers. And the way this muscle works and the way it interacts with the lens is really quite ingenious. As this muscle contracts, it makes the aperture smaller that surrounds this lens. And as this muscle contracts, then the tension on these zonule fibers can relax. And consequently, active contraction of the ciliary muscle leads to a rounding up of the lens and an increase in its refractive power. When that muscle relaxes, then it returns to a wider aperture. That causes tension on the zonule fibers. And as a result of that tension, then this lens is pulled more towards a flattened configuration. So one other biomechanical element here that makes all this work is the lens itself. So the lens will tend to ball up into a rounder configuration if it were to be cut free of those zonule fibers. So the elasticity of the lens itself is what gives it the capacity to achieve this more rounded configuration, which is appropriate for near vision. Now, adjustments to the shape of the lens are not the whole story when it comes to accommodation. There's also adjustments to the pupil itself. So the pupil is basically the space created by the iris. So here you notice the iris is a muscular structure that creates this space in the middle. And that is the dark spot that you see when you look deeply into the eyes of another person. Well, of course, what we're looking at is darkness in the interior of the back of the eye. The iris performs this very important function of minimizing optical aberration. Now, the iris also serves to achieve that optimal balance between the amount of light that's necessary to activate the neural elements in the back of the eye called the retina, and overactivating them or bleaching them. So we have an exquisite mechanism that allows inputs from the retina to drive circuitry in the brainstem to constrict that muscle and make moment-to-moment adjustments in the diameter of the pupil. This is called the pupillary light reflex, and we'll spend a bit of time talking about that circuitry in a different tutorial. Well, I think we're ready now to consider those neural elements at the back of the eye, and we will spend much of the rest of the tutorial talking about those elements. What I'm referring to, of course, is the retina. Now, I want to make an important embryological point as we begin to consider the retina, and that is that the retina is really considered to be truly part of the brain. And the reason is that the retina is diencephalic, which is to say, the retina is derived from the developing forebrain, specifically the diencephalon. So as the diencephalon begins to differentiate at the posterior part of the prosencephalon, something called the optic vesicle forms. And this is an outward pouch that we see from the lateral margins of the diencephalon. And this pouch grows out to meet other tissues that together form the eye. The retina and an important layer of tissue just behind it called the pigmented epithelium are derived from this diencephalic tissue. So one might wonder, why then do we call the optic nerve if it's really an extension of the brain? Well, I think that's a legitimate question. But the important point I want you to take away here is that the retina provides us with a unique opportunity to look into the eye of a patient and actually see tissue derived from the brain. And that gives us an opportunity to examine that tissue as well as the vascular supply to it and perhaps gain some insight as to the status of what's going on deeper in the cranium. All right, well, let's look now at the retina itself. And as I mentioned, there are two kinds of tissues here that are derived from this optic cup that constitute the retina. Right at the back of the eye is a tissue called the pigmented epithelium. This is a very dark tissue because of the presence of a pigment, and I'll talk more about that particular layer of cells in just a moment. The rest of this is what we call the neural retina, and it's comprised of 5 basic types of cells. And I want you to notice something about the organization of those cells relative to the eye itself. So the back of the eye is out here somewhere. Okay, so the pigmented epithelium is against the inner surface of the sclera, which is what we call the white part of the eyeball that we see from the outside. And in order for light to activate the cells that do the job of phototransduction, it has to pass through this entire neural retina because the phototransduction process is out here, right up against the pigmented epithelium. So this is where phototransduction happens. And that may seem somewhat surprising, that light would have to pass through this tissue before striking the photopigments that transduce that energy and generate an electrical signal. But we think this is the way it is for a very important reason, and it has to do with the relationship between these photoreceptors and this pigmented epithelium. So if we look more closely at that relationship, what we discover is that the outer tips of our photoreceptors make intimate contact with this pigmented epithelium. And that's critical for the function of the pigmented epithelium in the whole process of keeping these photoreceptors competent to transduce photon energy into neural signals. The pigmented epithelium is involved in recycling the discs of membrane that contain the photopigments. And we know that from experiments that allow us to trace the movement of discs from a more proximal position to a more distal position. And what we find is that eventually the discs are completely engulfed by this pigmented epithelium. And that's important in recycling the photopigment that's otherwise bleached by the interaction with photons. So this raises a rather serious problem that can happen with trauma, and that is what can happen when the photoreceptors are detached from this pigmented epithelium. So this can happen with a sharp blow to the head. It's a particular problem in people who are in sports that are subject to a blow to the head, and it's certainly something that can happen with a motor vehicle accident or some other trauma to the head. So retinal detachment prevents this intimate relationship between pigmented epithelium and photoreceptor, and as a result the photoreceptor no longer has a chance to renew the photopigment and recycle its disc membrane. So now let's consider the rest of the cells that comprise the neural retina. So the retina is made up of 5 basic types of cells. We've already talked some about the photoreceptors, and they're found in these sublayers of the retina from distal to proximal. There's the outer segment, which is where we find the discs that contain the photopigment. There is a layer called the outer nuclear layer, which contains the cell bodies of these photoreceptors. And then there is a layer of synapses called the outer plexiform layer. This is where the synaptic terminals of photoreceptors interact with 2 of the remaining 5 types of cells that we have in the retina. These synaptic terminals of photoreceptors release neurotransmitter on the endings of a type of cell called the bipolar cell. Here we have an illustration of a bipolar cell. Its cell body sits in the inner nuclear layer, and it's called bipolar because it extends processes in 2 directions. In the distal direction, it makes contact with the photoreceptor terminal. In the proximal direction, it makes contact with 2 of the remaining cell types in the retina. So in addition to the bipolar cell, we also have in the outer portion of the retina a cell called the horizontal cell. And it's called a horizontal cell because it gives rise to processes that extend over some distance in the horizontal axis of the retina. The horizontal cell both receives input from the photoreceptor, but also gives rise to input to the photoreceptor terminal. So there's a bidirectional relationship from a physiological perspective between the photoreceptor and the horizontal cell. Well, let's look in the inner part of the retina and encounter the 2 remaining cells that we find. As I mentioned, the bipolar cell sends signals that interact with these 2 cells, and the major cell that we find in the inner part of the retina is called the retinal ganglion cell. Now, just a word about this name. It may be confusing to you that we use the word ganglion to describe this particular cell. We're used to thinking about a ganglion as a nice ball of cells that sits outside of the central nervous system. Here, rather than having a ball of cells, what we discover is that the ganglion cells constitute a layer, several cells thick, across the inner surface of the retina. Well, the ganglion cells receive direct input from the bipolar cells. They also receive indirect input from the amacrine cells. And the amacrine cells, like the horizontal cells, give rise to horizontal connections that extend some distance across what's called the inner plexiform layer of the retina. So I want you to notice that in the histology of the retina, we have this alternating nuclear layer, plexiform layer, nuclear layer, plexiform layer, and then really another nuclear layer that we call the ganglion cell layer. And it's here that we find our 5 principal cells and their connections. Well, I'm not sure how much background noise you're hearing right now. I'm rather quite enjoying the sound of the rustle of the leaves and the birds and the cawing of the crows, but I'm quite irritated at the moment because it sounds like I hear the canned music of an ice cream truck. I don't know if you experience that phenomenon where you are, but we seem to have a vehicle driving not too far away, playing music, trying to attract people to sell ice cream. Well, hopefully that vehicle is on its way, gone, and we can return to our discussion of the retina. Okay, I need to make a few additional points about these cells in the retina. Let me point out how short these distances are in the retina. The entire thickness of the human retina is not more than a millimeter or so. And as a result, there really is no need to generate action potentials to get a signal from, let's say, the outer nuclear layer to the internuclear layer, or from the internuclear layer to the ganglion cell layer. However, it's quite a different story when we're talking about getting signals from the retina back into the brain. So as a result, what we've discovered electrophysiologically is that photoreceptors generate graded potentials. Bipolar cells generate graded potentials, but ganglion cells, they generate action potentials. And we think this makes good sense because the graded potentials are sufficient to span the shorter distance from one cell to the next within the retina. But if the ganglion cell has any hope of sending an electrical signal on its way to the brain, then action potentials are going to be needed to traverse that distance with fidelity. And indeed, that's the case. So the ganglion cell is the only neuron in the retina that generates an action potential. Well, there's one other cell worth mentioning here before we move on, and that is a special type of glial cell, which is not illustrated here, but glial cells exist in the retina, and this cell is called a Mueller cell, and it functions in a variety of ways to support both the metabolism and the signaling activity of the neurons of the retina. Well, now I'd like to focus in on the photoreceptors and the phenomenon of phototransduction. As I mentioned when we began to consider our sensory systems, the challenge of phototransduction is one that needs to be explained and accounted for in each of our sensory systems. And for vision, just speaking for myself, I always consider this such a deep mystery. I remember as a child just wondering how could it possibly be that we can see? How is it that this energy that we now understand to come in the form of particles and waves of light, how can that energy turn itself into signals in our brains that give rise to perceptions? Well, this is really a profound question, and we don't know the full answer yet, but we've gained considerable insight in the last several decades about how the process of phototransduction works in the photoreceptors. So let's turn our attention to these photoreceptors. What we find is that fundamentally we have two types of photoreceptors in the retina. There's a type of cell called the rod, and the rod is so named because of its long cylindrical outer segment. And there is a cell type called the cone. And rather than having a rod-shaped outer segment, it has more of a conical-shaped outer segment. Well, there are multiple differences that we want to discuss concerning these two cells, and we'll get to that shortly. But first, I want to focus in on the issue of phototransduction, which we know best for the outer segments of the rod. And first, let's consider what happens in the outer segment of the cell when it's kept in the dark. And the surprising feature of this photoreceptor in the dark is that it's actually depolarized, at least relative to other neurons that we've talked about so far in this course. And this depolarization is supported by something that we call the dark current. In the plasma membrane of the outer segment of our photoreceptors, we have an ion channel that is permeable to calcium and sodium. This channel is gated by the second messenger, cyclic GMP. So when cyclic GMP is present in abundance, it interacts with the cytoplasmic face of the channel, This allows calcium and sodium to diffuse down their concentration gradients and enter that postsynaptic cell. Well, this helps to support a depolarized resting membrane in these cells. There are other ion channels in the photoreceptor that allow for the efflux of potassium. Well, when a cell is in the dark, the influx of calcium and sodium contributes significantly to the resting membrane potential. So for this type of cell, we have to consider these additional ions if we're going to predict the resting membrane potential. Unlike our simple neurons and axons that we considered earlier in the course, where a leak for potassium was sufficient to approximate the resting membrane state. Here, we have to also consider these additional active conductances for calcium and for sodium. Now, the process of phototransduction requires the interaction of a photon of light with a photopigment. And the photopigment is a complex molecule which consists of a opsin, which is a large protein structure that interacts with a smaller organic molecule called 11-cis-retinal. Now, this opsin is actually going to be familiar to you because it is a member of this 7-transmembrane protein family that we've encountered previously in this course. We've encountered this general structure previously. The 7-transmembrane structure is exactly what we saw when we considered metabotropic receptors for neurotransmitters on postsynaptic terminals. In fact, the opsins are part of that same family of proteins. Quite remarkable, don't you think, that a metabotropic structure is present both in postsynaptic membranes within the brain and as a key component of the photopigment in the eye? Well, let me tell you about the role of these two components. So the 11-cis-retinal is the organic molecule that will interact with the photon of light directly. The opsin that surrounds that 11-cis-retinal fine-tunes the response profile of that 11-cis-retinal. So we actually have 4 kinds of pigments in our photoreceptors. In rods, we have the rod opsin, and it has a particular spectral sensitivity, which is plotted in this chart of relative spectral absorbance versus wavelength of light by this dashed line here. And you can see it's somewhere near the middle among our family of opsin proteins. Cones contain one of 3 different opsins, and those opsins allow for the fine-tuning of the responses of the 11-cis-retinal to either shorter wavelengths of light, medium wavelengths of light, or longer wavelengths of light. And obviously the medium and the long are much closer in their spectral sensitivity than they are to the short. So these 3 different opsin proteins that are found within the cones provide the foundation for color vision. We'll say a little bit more about color vision as we move on in our studies of vision. Well, let's see what happens when light interacts with this photopigment. Here we have a recording of membrane potential from a photoreceptor, and the experiment is to provide a very brief flash of light. And what we can find is that there is a graded response with the intensity of that flash of light. But notice the polarity of the response. Again, the resting membrane in the photoreceptor is around minus 40 millivolts, and the response to light striking the outer segment of the photoreceptor is hyperpolarization. And with increasing intensity of light, we see a deeper hyperpolarization, perhaps down to about minus 65 millivolts. Okay, well, how does this work? Well, when light strikes the outer segments of the photoreceptor, there is a series of molecular events that lead to a reduction in the concentration of cyclic GMP. So when the level of cyclic GMP is reduced, then these channels close. And the closure of these cyclic GMP-gated channels means that calcium and sodium can no longer enter this cell and support its depolarized state. Rather, the potassium channels remain open, allowing for the efflux of potassium, and that brings that membrane down to a more hyperpolarized state. And that allows the membrane to hyperpolarize to a predictable equilibrium near about minus 65 millivolts for many photoreceptors. Well, let's look now in a little bit more detail at the molecular level as to how this happens. Well, we have our 11-cis retinal that is in the center of the opsin, and when light strikes this molecule, there's a rotational twist between carbon atoms 11 and 12. That converts the 11-cis retinal into an all-trans configuration. So you may remember a bit of organic chemistry there, the difference between a cis and a trans. Well, this conformational shift is what leads to the activation of the metabotropic element of the opsin protein. So with that in mind, here is light striking our photopigment, and the twist that we see in that retinal molecule leads to the activation of a set of G proteins that are affiliated with that opsin. Those G proteins are called transducin, and one consequence of activating these G proteins is that a phosphodiesterase enzyme is turned on, and that phosphodiesterase cleaves the cyclic structure of the cyclic GMP molecule. And as a consequence then, the cyclic GMP levels fall, and the channel goes from its open configuration to its closed configuration. Rather remarkable that all of this happens as quickly as it does. Well, in neural terms, it actually does take a bit of time, which contributes to a bit of the lag in the operation of the visual system. The visual system is actually one of our slowest sensory systems, in large measure because of the time necessary for this phototransduction cascade to play out. Well, now let's consider some features of rods and cones that help to differentiate their contributions to vision. Rods are extremely sensitive to light, for reasons that we'll mention in just a moment. Cones, on the other hand, are relatively insensitive to light. Vision, when driven by rods, is characterized by low spatial resolution, and this pertains to the wiring of rods to ganglion cells in the retina. Cones, on the other hand, are associated with very high spatial resolution. Putting these facts together, rods are specialized for very low light levels. We call this scotopic vision, whereas cones are specialized for very bright conditions, such as this beautiful late afternoon here in North Carolina where I'm talking to you today. So this photopic vision enables color vision, since color vision requires the activation of cones. Well, let's consider some of the physiology and anatomy of our rods and cones that helps to explain these features. If we look at the response of an individual rod and a cone to the onset of light, what we find is that in a rod, there's a graded response, graded by the intensity of the light stimulus, but there's also a very sustained response that adapts relatively slowly. Now contrast that to the responses of the cone, which also responds in a graded fashion, but the adaptation is very quick. So the metabolic mechanisms that are responsible for adaptation within the outer segment of these cells operate on different timescales, with a much more sustained response seen in rods relative to cones. Another specialization pertains to the wiring of rods and cones to the ganglion cells, and this is shown rather schematically here in this figure. Notice that about 15 to 30 rods connect up with a single bipolar cell. Contrast that to the cone system, given this one-to-one relationship, and this cone bipolar cell can be sensitive to a much more narrow spatial channel as photons interact with the retina. We can consider the specializations of rods and cones in terms of their physiology and their anatomy and begin to understand how we can be sensitive to light over the incredible range of luminance that's present in our environment. We're sensitive to more than a 12 log unit range in luminance. We can define an absolute threshold for vision, and this occurs under extremely dark conditions. Imagine being in the darkest woods in the darkest night and your way illuminated only by starlight. What little vision that's possible under those conditions is due entirely to the operation of our rod system. This is called scotopic vision. Now, in conditions such as where I find myself now, looking into the setting sun, there is considerably more photons in this environment right now. And I suspect that I'm somewhere around this amount of illumination now, where our cones are responsible for the vision that I see. The rod system has completely saturated at this point. So all that I see in my world around me at this moment is due to the activation of cones in my retinas. So these kinds of conditions, when cone vision dominates, are called photopic. Now there is a transitional region in the middle called mesopic illumination, and this would be characterized by, let's say, being out in the woods, and in addition to starlight now there's moonlight, maybe about a quarter moon or more. That would get us in this range where we are beginning to approach photopic conditions that are possible when there's nearly a full moon. So this operating range is incredible, and it is a real feat of evolution that we can remain sensitive to the dynamics of light in our environment across such a huge range in the physical world. And that has to do with the responses of the neural elements and how they interact with one another to actually produce the signals that are conveyed from the retina to the brain. Well, one final specialization to consider is the distribution of rods and cones across the retina. Well, I've already mentioned earlier that in the center of our vision we have a very high density of cones, and here's what this looks like. So what we have in the plot here down below is the distribution of cones, shown in green and very low levels in the peripheral parts of the retina. But as we approach the very center of our gaze, we find this very interesting specialization in the retina called the fovea. And in the fovea, we have the complete exclusion of rods. And what we see is a very high density packing of cones with minimal diameter to the outer segments of those cones, making them as sensitive as they possibly can be with respect to the spatial arrangement of the distribution of photons that might be striking that retina. As the cone system gets further removed from that center of vision, notice that there are far fewer cones that are present, and they tend to be much larger as we get out. Now contrast that with the rod system, and what we find are that the concentration of rods increases as we get near the center of the retina, but then it falls precipitously as we approach the fovea to a total area of exclusion. And then it rises again, only to trail off as we approach the peripheral parts of the retina, where the density of all photoreceptors begins to trail. So this fovea then is quite an interesting spot in the retina. Let's take a closer look at it. If we looked at the architecture of the connections between photoreceptors and ganglion cells in the fovea, what we discover is that there is a pit in the middle of our fovea. And fovea itself means pit, but that central region is called the foveola. And in that foveola, we have a vascular free zone where the blood vessels themselves are removed from the path of light striking the photoreceptor. Not only are the blood vessels removed, but the neural elements of the retina themselves are pulled apart to the side, allowing for nearly a direct access of photons to the photoreceptors and their outer segments where the photopigments reside. So, although light can pass through these neural elements without too much of a decrement, evidently in the center of the fovea, there's some additional cost that is invested to clear away those neural elements, allowing for maximal efficiency of phototransduction. Now, as this illustration should make clear, this is a very important part of the retina for vision. This is a part of a retina that can degenerate with age and with certain genetic and perhaps dietary or environmental factors that interact with our genetic constitution. And what I'm referring to is the degeneration of this part of the retina that's also called the macula. That means spot or yellow spot, macula lutea. So the macula is at the center of our retina, and the fovea is at the center of the macula. So when this degenerates, we lose the ability to see right in this central part of vision. And if I can return to that scene that I showed you earlier, this beautiful scene in Lake Louise in Alberta, Canada, if the viewer now were to have macular degeneration, they might lose their capacity to see exactly what they might be interested in detecting. If they wanted to look at our faces, it would be very frustrating because exactly their direction of gaze is what would be most affected with macular degeneration. Well, now let's turn our attention to the structure of receptive fields of the ganglion cells. Again, these are the cells that generate the action potentials that send signals from the retina to the brain. We talked about receptive fields when we introduced them in the somatic sensory system, and the definition of the receptive field that we gave was a region of the body that, when stimulated, can modulate the activity of nerve cells. Well, the same basic principle can apply to our retina, only now the stimulation of the body would be the retina itself, a patch of photoreceptors that would modulate the activity of the ganglion cell. Well, because that stimulation is coming from light in the environment, we can also define the receptive field in terms of a location in the visual environment that, when illuminated or when there's some change in the level of illumination, can lead to a modulation in the firing of retinal ganglion cells. One additional point about receptive fields for ganglion cells in the retina is that they also bear the center surround structure that we saw in the somatic sensory system. That is, there is a central region that is especially sensitive to changes in luminance, and then there's a surrounding region that has an antagonistic relationship with the center. Well, putting these things together, we can recognize that there are these two kinds of ganglion cells that have different center-surround configurations. There's the on-center ganglion cell that responds best when light strikes the center of its receptive field, and then there is the off-center ganglion cell that responds best when light strikes the surround. So let's see how these two cells would respond when we illuminate the center of their receptive fields. So at time t1, a spot of light goes off that illuminates the center, and what we see is a nice barrage of action potentials in our on-center ganglion cell. Meanwhile, in our off-center ganglion cell, what activity might have been present in the dark condition goes away. And when the spot of light in the center goes off, now there's a bit of a rebound discharge. So we have an increase in action potentials when the light goes off. Now, because we're talking about light and we can alter the level of light, we can imagine now decreasing the amount of illumination to the center of the receptive field. And when that happens, the on-center cell decreases its firing, and when that dark spot is removed, or essentially there's now an increase in illumination to the center, there's this nice rebound discharge. So that's the on-center. Now let's look at the off-center cell. Off ganglion cell, then there is an increase in discharge. And when you return to that higher level of illumination, then this cell becomes less active. Well, this may seem a bit curious, but putting these two classes of ganglion cells together, we have cells that might be sitting right next to each other in the retina. One member of the pair will increase its firing when light goes on, and the other will decrease its firing. But light can go off, and when that happens, we see the converse. The on-center cell will decrease its firing while the off-center cell will increase its firing. Putting these two kinds of cells together makes each location in the retina sensitive to changes in the levels of light, be they increases or decreases. Now, one additional consequence of the center surround organization I should mention is shown here in the final panel, panel C. When we evenly illuminate both the center and the surround, notice that there's not nearly so much change in the firing patterns of these cells. While the small spot in the center gave rise to a nice robust discharge, a more even illumination across the receptive field gives rise to a much lower level of activity in our on-center cell. And the same sorts of relationships would be seen for the off-center cell. The off-center cell stops firing with the presence of light in its center, and with more even illumination, it now begins to fire just a little bit, but not as much as when the dark field is once again returned, when the light goes off. Well, let's look at this behavior in a different way. So now imagine that we have an on-center cell, and we have a small spot of light that is passing across the receptive field from right to left. As we pass that spot of light from the center of the receptive field, we notice that there is initially a pretty strong response in the firing rate of this on-center ganglion cell. But as the spot of light acquires an increasingly more peripheral position, notice that the response falls, and when we are illuminating the surrounding part of the receptive field, there's actually a decrease below spontaneous level in firing. So this is the classic behavior of the on-center ganglion cell that I just described. How do these center surround receptive fields really contribute to visual perception? Well, there's still a lot of work addressing the possible answers to that question, but one provisional answer seems to be that this center surround structure gives us the ability to be sensitive to edges of light. Now imagine, rather than a small spot of light going across your retina that perhaps might have been a satellite that you might be looking up into the night sky and seeing a satellite passing overhead. Now imagine the much more common experience of having light and shadow in more of a linear configuration falling across your retina. When that happens, we can see that the best response from this on-center ganglion cell is when there's shadow across a significant portion of its off-surround, while its center is completely on the illuminated side of this boundary. So when there's a luminance boundary, when there's light and shadow, and that edge is completely aligned with the center surround boundary, then we have the greatest modulation of activity coming out of that particular cell. Well, we're getting close to the end of this tutorial. There's a few more things to be said about the organization of these ganglion cell receptive fields. I want to address why we have on and off center responses in the first place. It does seem a bit odd that we should have these dichotomously arranged ganglion cells that respond in exactly opposite ways to the very same stimulus. Well, how is that possible? So the neurotransmitter that's released by the bipolar cell is glutamate. And the receptors that are expressed on these two different types of bipolar cells are quite distinct. In the on-center bipolar cell, the receptor is a metabotropic receptor for glutamate. And that metabotropic cascade that's initiated results in the hyperpolarization of that bipolar cell. For the off-center bipolar cell, the receptors are AMPA receptors. And when glutamate binds to the AMPA receptor, there's a depolarizing response. So this is what's happening in the dark when the cell is depolarized at the level of about minus 40 millivolts and glutamate is being released constantly in the dark. It's keeping that on-center bipolar cell hyperpolarized, meaning that it is not releasing its neurotransmitter on the on-center ganglion cell. So the on-center ganglion cell is essentially turned off in the dark. Meanwhile, glutamate being released and interacting with the off-bipolar cell is going to lead to the depolarization of that off-bipolar cell, the release of its own transmitter, and the activation then of the off cell. So different receptors for the same transmitter provides the basis for our on and off responses in the center of the receptive fields. So now let's consider what happens when light strikes the center of the photoreceptor receptive fields that are driving activity in these two parallel channels. So recall that when light strikes the photoreceptor, there's hyperpolarization. Hyperpolarization means much less release of glutamate, the neurotransmitter of the photoreceptor. When we release less glutamate, then the on-center bipolar cell is going to depolarize. Remember, the metabotropic receptor mediates hyperpolarization. So if you withdraw the transmitter that's interacting with that receptor, then this cell is going to depolarize. It will depolarize, it will release its neurotransmitter, which leads to the activation of this on-center ganglion cell. And that cell now responds to the illumination of the center of its receptive field. Now let's think about what happens to the off-center bipolar cell. So when there's less glutamate being released, that means that there's going to be less glutamate to interact with the NMDA receptor, and that's going to lead to the hyperpolarization of this off-bipolar cell. If there's hyperpolarization, then there's going to be less excitatory neurotransmitter released, and that's going to have a suppressive effect on this off-center ganglion cell. Again, the key principle here is that we have two very different receptor systems available for the neurotransmitter of the photoreceptor. And those different receptors set up these dichotomous responses in these bipolar channels to the ganglion cells. So now let's consider what happens when we decrease the illumination of the center of the receptive field. If we do that, then we imagine that we are withdrawing photons from that photoreceptor, allowing it then to depolarize even more than where it was with background illumination. So now with greater depolarization, there's going to be an increase in the release of synaptic glutamate from the terminals of that photoreceptor. So with an increase in synaptic glutamate, the on-center bipolar cell is going to hyperpolarize due to the activity of the metabotropic receptor system, whereas the off-center bipolar cell is going to depolarize because glutamate's activating its AMPA receptors. And as a consequence, the off-center cell is going to fire a barrage of action potentials with this decrease in light level of the center. Meanwhile, the on-center cell is going to be suppressed. Well, there's one more issue to consider here with respect to the organization of the receptive fields of ganglion cells, and that is the center-surround interaction. Well, I'm not going to discuss in detail how all of this works. What I'll rather simply say is that the horizontal cells are key to mediating the center-surround interaction. There is a bidirectional signal that is antagonistic, or we call this a sign-inverting signal. The glutamate that's released by the photoreceptor activates horizontal cells, which in turn release GABA on the terminal of the photoreceptor as well as surrounding terminals over some considerable spatial extent of the retina. And this antagonistic interaction between the photoreceptor and the horizontal cell is what gives rise to this antagonistic relationship between center and surround, with the surround signal suppressing the center signal. Well, one benefit of this center-surround antagonism is that what these ganglion cells really convey to the brain is a signal not so much about the overall illumination of the environment, but rather about contrast, about an edge of light that might just be perfectly dividing the center from the surround region for any one receptive field structure. Well, with this sensitivity to contrast, the retina now can respond over a tremendous range of environmental conditions. What we're looking at here is a plot that illustrates the dynamic range of the response of a ganglion cell, and each of these lines here represent the discharge rate of a ganglion cell over some range of test spot illumination in the center. The different lines correspond to these different amounts of background illumination. And the point here is that a ganglion cell can respond to more than 6 log units of illumination, depending upon the amount of contrast that's present between the center and the surround. This allows vision to happen under a remarkably broad range of environmental conditions. Just as I've been speaking to you in this tutorial, the sun has set and the environmental conditions are much less bright now than they were at the start of this tutorial. Believe it or not, if I were to look at a piece of print, the light reflected from the white paper would be less than the light reflected from the dark print at the start of this tutorial. So what appeared to be dark when the sun was still up still looks dark to me now, even though the amount of light that's being reflected is actually considerably less. And similarly, the white paper still looks white, even though it's now dusky and the sun is going down. So that's just one example of the functions that are illustrated here in this curve, where what our retinas are really conveying to the brain is contrast, defined by edges of light and shadow. Well, I want to show you one example of this, and for this I'm going to pause just a moment and navigate over to the website of a good colleague of mine and friend and mentor, Dr. Dale Purvis, who has one of the best websites that pertains to any field of science available on the World Wide Web. And I would invite you to navigate there yourself. His website is purvislab.net. Well, I've just landed on the website of Dr. Dale Purvis, who you'll recognize as being the senior author and senior editor of the textbook that I've been referring to. Again, a good friend and colleague for many years now here at Duke University, and a mentor of mine throughout the last couple of decades of my career. So I'm going to click on the domain in his website called See for Yourself. And if we go to See for Yourself, I want to draw your attention to one particular animation here under the lightness and brightness category. Now hopefully you can see this dot that's moving back and forth. It's a circular patch, and I hope that as you watch it move, you'll be able to see that there's an apparent change in the brightness of this circular patch as it passes through what looks like a bit of a veil or a bit of a fog. As it moves to the right, we see that it appears to be dark, and then as it moves to the left, it appears to be bright. So there, now it's getting brighter, and then as it moves back to the right, it's getting darker. I hope that's exactly what you see. So here it's getting bright, now it's getting dark. Well, I just want to see if you're paying attention. Of course, that's not what I see, and I hope that's not what you see. As it moves to the left, it's getting dark. You see that? It's getting dark. Now it moves back to the right. It's getting bright. To the left, it's getting dark. To the right, it's getting bright. Well, this is a demonstration of exactly the functions that I showed you in the last slide. So there's a change in the background illumination, and as that happens, our visual system continues to be responsive to the contrast between the center and the surround. Now, if you read a little bit more about what Dr. Purvis has to say about this demonstration, he'll tell you that the physiology of retinal ganglion cells just can't possibly explain this phenomenon. There must be something deeper in how these signals are processed in the brain that gives rise to this percept. And he very well may be correct about that. But all of this begins in the retina, and the physiology of the receptive fields of the ganglion cells has much to say about our ability to detect this patch of gray as it moves across this background illumination. Now, in case any of you have doubted whether this is actually a gray spot, this website has this nice little interactive feature. So here I can just mask out the background and move this with the spot, and you can see that indeed it's a uniform gray spot that moves across this field. And we can let that pass out and see how indeed the luminance is a function of the interaction of the spot and the background. Well, we could spend quite a bit of time here on this website, and I would invite you to do so. I think it's a fascinating set of demonstrations of visual physiology, and we'll look at a few more in our next tutorial. Welcome to this tutorial on the visual system, and specifically I'd like to talk to you in this session on central visual processing. We're going to talk about some of the most wondrous and most interesting aspects of cortical physiology that will highlight once again the complexity of the brain among the body's organs. We will encounter some circuitry that's genetically determined, and that circuitry will form the foundation for visual processing in the brain. And some of the topics we will consider today are just things that just fill me with a sense of awe and wonder at what the human brain can accomplish. And that sense of wonderment is what drives my own personal curiosity to try to understand how the brain works. I have several learning objectives for you today. I want you to be able to describe the distribution of the axons of retinal ganglion cells to the major processing centers in the forebrain and the brainstem. And then I want to focus on what's going on at the level of the forebrain, specifically in the primary visual cortex. I want you to be able to discuss the major receptive field properties that we discover when we record electrical signals from neurons in V1. I want you to be able to discuss in very general terms the functions associated with those parts of the visual cortex that are beyond the primary area. We call those extrastriate visual pathways. All right, let's once again consider the distribution of the axons of retinal ganglion cells. So the ganglion cells of the nasal retina send their axons through the optic nerve. They cross the midline and project to targets in the diencephalon and in the midbrain on the opposite side of the brain. Those retinal ganglion cells from the temporal retina send axons that grow through the optic nerve, and as they approach the chiasm, they remain on the ipsilateral side of the brain. And as a consequence, once we get visual signals posterior to the optic chiasm, now each optic tract contains all the information about the opposite side of the visual world that's seen by each eye. Now, the major target of this visual information with respect to the image-forming aspects of vision is the lateral geniculate nucleus. And we see that nucleus illustrated here in the posterior and inferior and lateral aspect of the thalamus. It's the major target of retinal ganglion cells with respect to vision or image formation. And from the lateral geniculate nucleus, arises are our optic radiations that work their way around the lateral ventricle to supply the upper and lower banks of the calcarine sulcus. That's where we find our primary visual cortex. Now, here to the right is a histological view of the primary visual cortex that was generated by a good friend and colleague of mine back in the day when we were both working as postdoctoral fellows together. And this is a beautiful histological view of a myelin stain through the cortex that forms the calcarine sulcus. So here in this space, here's the calcarine sulcus as labeled there. And you'll notice if we look into the thickness of the cortex with the myelin stain that there's this striking dense band of myelination that runs right through the middle of the cortex. And one feature of this band that is especially striking is how it so clearly defines a region of cortex here. One can recognize exactly where this band has its terminations. And the early anatomists, such as Corbity and Brodmann, looked at this part of the cortex and concluded that the presence of this stripe, which is called the stria of Gennari, must signify that there's something distinct about this bit of the cerebral cortex. So Brodmann labeled this Brodmann's area 17, or that's how we know it today. He just called it area 17. And it's also known as the striate cortex because of this histological presentation of what we call the stria of Gennari. Now, one interesting aspect in the history of neuroscience over the last 5 or 6 decades or so is that probably no single area of the brain has attracted more interest, more attention than this striate cortex. And one reason is that it's within the circuitry of the striate cortex that we come best to understand this computational function of cortical microcircuits. You will recall, I trust, what I suggested is an easy way to remember the function of the cortical microcircuitry, the ACC of the cortical column. The A stands for amplification of the thalamic input. The C, the first C, stands for computation. That is the emergence of new functional properties that weren't present in the input signal that is amplified by this cortical circuitry. We'll see some wonderful examples of computational properties that emerge as a result of the operation of cortical networks. And then the final C is communication, as these signals are then disseminated along higher order pathways. We'll see an example of that here as well. So the ACC of the canonical cortical microcircuit was truly first established by studies of this part of the brain, the visual cortex. Two of the leading investigators that has made all of this possible are David Hubel and Torsten Wiesel, who very deservingly were awarded the Nobel Prize in Physiology or Medicine in 1981 for their foundational work on the physiology, the anatomy, and the developmental plasticity of this part of the brain. It's really difficult to overstate the influence of these two scientists on the development of modern neuroscience. There were others at the time that were beginning to eavesdrop on the nature of neural signals in the cerebral cortex. Most notably at that time was Vernon Mountcastle, who was studying cortical columns in the somatic sensory parts of the brain. But for the visual system, it's really David Hubel and Torsten Wiesel that paved the way for generations of neuroscientists who would follow, including myself, who have been fascinated with trying to understand the physiology of the circuits that arise in this part of the brain and how they might be shaped by our visual experiences in early life as we open our eyes and begin to encounter the world. Well, I could go on about Hubel and Wiesel and their influence over my career and the career of many of my colleagues, but we will press on and turn our attention to some of the receptive field properties that Hubel and Wiesel discovered in this part of the brain. And I'd like to share a little bit from my own research lab some of the ways that we are continuing to build upon what Hubel and Wiesel taught us and hopefully extend our understanding of this fascinating part of the brain. Now, as we get into this discussion, let me remind you that at earlier stages of visual processing, such as in the retina and even in the lateral geniculate nucleus, cells respond best to small spots of light. They have receptive fields that are organized into center and surrounding regions that are circularly symmetrical. And so what we see in the thalamus is very much similar to what we saw in the retina. There's no major transformation of the nature of visual representation from retina to thalamus. However, if we record from cells in the visual cortex and try to stimulate with the same stimuli that works so well for retinal ganglion cells or lateral geniculate nucleus neurons, we see very subtle and not terribly robust responses. But rather, if we change the stimulus, if we replace the small spots of light with large edges of light or bars of light, and then move those bars in one direction or another at one orientation or another, let's say horizontal or vertical, then we are very likely to record robust responses in the primary visual cortex. And this is exactly what Hubel and Wiesel did. And what they discovered are two very important computational properties of this cortical network: orientation selectivity and direction selectivity. Let me illustrate what they found by considering cortical orientation selectivity. So Hubel and Wiesel did experiments such as this using animal models, and this highlights the importance of animal models in modern neuroscience. Of course, we are highly concerned for the welfare of all animals that are used in science or those animals that are kept as pets by private individuals as well. We need to treat these animals with the highest standards of humane concern for their welfare, and that's certainly the case, generally speaking, in the field of neuroscience. I know in my own work with animals and research, the standard is to keep these animals as comfortable and as healthy as we possibly can. Keep the animals socially engaged, supported in their nutrition and in their physical activity. And in the conduct of the experiments, the goal is to eliminate the experience of pain. So consequently, when experiments are done on animals, they are anesthetized before any intervention is done. And while the experiments are ongoing, the animals are very carefully monitored to be certain that they are appropriately anesthetized in order to ensure that there is no pain experienced by the animal. And in the case of terminal experiments, at the conclusion of the study, the animal is simply put down with an overdose of anesthetic. Well, this is the kind of experiment that was done by Hubel and Wiesel using cats and young kittens. And an illustration of one such experiment is shown here. We have an animal that's been anesthetized and prepared for surgery, and it's anesthetized to the point where it's feeling no pain, but not so deeply that there's no brain activity at all. Otherwise, of course, that would be incompatible with life. So the animal is alive, but it's deeply anesthetized, much like a person undergoing surgery. Well, a microelectrode is implanted in the visual part of the brain of this animal, back in the occipital cortex. And from that microelectrode, action potentials are recorded from neurons while these animals view light stimuli that are projected on a screen in front of the animal. As I mentioned, what Hubel and Wiesel discovered is that cells in this part of the brain respond especially well to a bar of light when it's moving in a particular axis of motion. And what they might even find is that some neurons respond very well when the bar is moved in one direction, but not at all when the bar is moved in the opposite direction. So these two properties together are what we refer to when we talk about orientation and direction selectivity. Orientation selectivity refers to the preferred response to a bar of a particular orientation versus another, and direction selectivity refers to the preferential response in a particular direction of motion, but not in the opposite direction. Okay, so here in the middle panel, we see a representation of data recorded from one such experiment, and the microelectrode tip happens to be sitting next to a cell that responds best to vertical stimuli. So when the preferred stimulus is presented, there's a robust discharge of action potentials from that cell. But as the orientation or as the angle of that stimulus is shifted away from vertical, the response gets less and less. After a series of such stimuli that are presented around the full cycle of possible orientations, it's possible to define a tuning function that plots the response of the neuron across a range of stimulus presentations. And for this neuron, what we find is that there is sharp tuning for angles near vertical. Now, there's not absolute tuning. There is some response off vertical, but by far the best response is within about 15 or so degrees of the preferred orientation, vertical in this case. Now, in the original experiments done by Hubel and Wiesel, and in many experiments done by many hundreds of workers since then, we know that if such a microelectrode is inserted into the cortex vertically, that is, in the radial axis of the cortical microcircuit, what we find is a series of neurons that we might encounter at different depths through the cerebral cortex that pretty much all report the same aspects of the visual stimulus. That is, if we plotted their spatial receptive fields, we find that all of these spatial receptive fields more or less overlap across the depth of this recording. And when we look and test for the orientation selectivity of these cells, we find that they are all pretty much sharing the same orientation preference. In this example, again, all preferring angles very much near 90 degrees. Now, a different story would be observed if the angle of the electrode penetration is made much steeper, and that's what's shown in this experiment. So now the microelectrode is coming into the cortex at a very sharp angle, such that we are passing across a set of cortical microcolumns, progressing from one to the next. And what we find when we move across the cortex in this way is that receptive field position slowly begins to drift from one location through adjacent locations until finally we reach a separation of a full receptive field after we advance a microelectrode about 1 millimeter or so in the cortical tissue. If we consider what happens with the advance of that microelectrode with respect to the orientation selectivity of the neurons, we find a similar shift. In one position, we may find neurons that prefer horizontal, and as we progress across the cortex, moving, let's say, in 100 micron increments, what we might see is a gradual progression of orientation preference as we move from one site to the next. And finally, we may discover that after movement of about 0.5 millimeter or so, that we might have a tuning function that is entirely non-overlapping with the first neuron recorded in this series across a penetration. So this suggests that there is a columnar organization of circuitry where all the neurons in the column tend to respond to the same stimulus, and that this columnar circuitry is organized in some systematic fashion, such that neighboring locations across the cortical surface are representing nearby aspects of the visual stimulus. And there is a smooth progression of change that occurs. Now, since the time that David Hubel and Torsten Wiesel did their experiments that earned them the Nobel Prize, additional methods have been developed, some of which allow neuroscientists to look at large-scale patterns of activity, not at the single cell level, but at the level of large populations of neurons over several millimeters of visual cortex. One such method is known as optical imaging of intrinsic signals, and it's based upon the fact that as brain regions become active, they draw oxygen off of the hemoglobin that's being supplied to local regions. That creates a bit of oxygen debt, and as a consequence, arterioles dilate and blood flow increases to those regions. And there's exquisite coupling between brain activity and blood supply that makes these signals a robust indicator of neural activity. Now, the method in question is called optical imaging because we don't need radioactivity as we would with PET imaging. We don't need a magnet as we would with MRI imaging. Rather, using experimental animals and in patients undergoing neurosurgical exposure of the brain, it's possible to directly illuminate the brain surface with light and record the subtle changes in the absorbance of light that happens as this oxygen and blood flow dynamic changes in brain tissue. So what you're looking at here is the results of an experiment that I actually performed. And in this study, we looked at the responses of the visual cortex in an experimental animal where the cortex is exposed to light and visual stimuli are presented on a screen in front of the anesthetized animal. So this panel that we have here in the upper right-hand side of the image is a view of the living brain. And all the little lines that you see, these are all blood vessels that are supplying the brain with the blood that it needs to be active. Now, if we take an image of the brain with no stimulus on the screen and compare that to an image taken while a stimulus is presented, we can subtract those images and look for the very small change in the absorbance of light that is a reflection of brain activity. When we do, we have the image that is shown here on the left. So we're looking at roughly about 8 millimeters by 6 millimeters of the visual cortex. And what we see are a series of dark spots and small bands. And these dark spots and bands are cortical columns that were activated by the stimulus. Now, it's interesting to look at the literature from the time of Hubel and Wiesel's original writings back in the 60s and 70s and see how even though they were probing the brain with microelectrodes, they still had a sense of the organization that this method makes now quite clear. And that is that there are columns of neurons that are responding to the same visual stimulus that are distributed across the visual cortex. And columns shift their preferences gradually from one location to the next, such that if we compared the responses, let's say, to a horizontal stimulus with those to, let's say, a vertical stimulus, what we would find is that the columns that are present in these two images precisely interdigitate. So the image we see here in the upper right is the result of the subtraction of the images acquired while the animal viewed horizontal stimuli and vertical stimuli. And it's a way of looking at how selective is the response. That is, how well can we differentiate the responses to one visual angle, horizontal, from another, vertical. And we can repeat this method over and over again with different kinds of stimuli, different pairs of orthogonal stimuli, and generate a family of images that reflect the interdigitation of columns that are representing orthogonal orientations. Now, there's a different way to represent these data that I'd like to show you here by looking now at a dynamic representation of a set of these images, where we will just loop around and see how the illusion of motion in these images gives us some insight into the organization of the functional map for orientation preference. What I'd like to show you are some data from my lab and the lab of my colleagues, where we've looked at different experimental animals and have measured the distribution of orientation columns in their brains. And we've created an animation that runs through all the images that were acquired as the orientation of the visual stimulus was changed. So if I start this animation and it will just run over and over. What you can see, I think, is this beautiful dynamic array of functional representation in the brain. Of course, it looks like something's moving in the brain. Nothing's moving in the brain. What is changing is the distribution of functional activity over time as the visual stimulus is rotated. And one point I'd like for you to appreciate is really how similar these patterns look in three very different species of animals. The tree shrew is a small animal that makes its home in Southeast Asia. The galago is a prosimian primate from Central and South America, and the ferret, of course, is a domesticated carnivore. So we're looking at the visual cortical patterns from animals that diverged about 65 million years ago, and yet there are very similar patterns and seemingly a highly conserved cortical network that's giving rise to these patterns. And if we look in even more detail, I find it interesting that we can localize certain locations in these images where it looks like these dark domains of activity are swirling around small points or singularities. And we call these points pinwheel centers. And it appears as though the organization of this representation of orientation preference is one of a large collection of pinwheel structures that are arrayed across the visual cortex. Well, one way that we can represent this kind of a dynamic set of experimental data in a static fashion is to perform a little bit of image math and then apply a color code, which allows us to show what we call the map of orientation preference. So we're looking at the very same region of the visual cortex in an experimental animal, and now we're using color to represent the preferred orientation at each location in the visual cortex. So for example, where we see the color red is representation of horizontal visual stimuli. So these are all cortical columns or bands of cortex that prefer horizontal among all possible orientations. And where we see blue regions in the brain, or rather brain regions that have been colorized blue, these are cortical columns that are representing near vertical. And between red and blue are all possible orientations that are represented in the brain. Now, the dark bands that we see, these are structures that we can't resolve the preference, but with newer techniques that allow us to see the activities of individual neurons in the living brain, we know that these zones are really not dark at all. Rather, they have cells that represent orientation just as robustly with as sharp tuning functions as any other place in the brain, but rather these cells with different preferences come together in very tight spaces. And with this method, we can't resolve their differences. But with a cellularly based method of resolution called two-photon imaging of calcium signals, it's possible to detect the preferences of neighboring neurons. Well, when we do so, what we discover is that there are locations such as right here, and there, for example, where there are point singularities around which a set of cortical columns are arrayed in a radially symmetrical fashion. And at that point singularity, nearby neurons have very different orientation preferences, such that we're looking at the center of one of these pinwheel formations. Well, I can go on and on about this as my colleagues and I have in our research. I won't belabor this point any further. But rather I wanted to give you a bit of an inside look at the circuitry and the physiology of this wonderful part of the mammalian brain, the primary visual cortex. And all of this has really just been to illustrate one computational property, and that is the property of orientation preference. Now, we and others have done experiments where we've plotted out the activity maps, not just of let's say, a horizontal bar moving along a particular axis of motion, but let's say a horizontal bar that's either drifting in just one direction or drifting in the opposite direction. And what we discover is that we may find a domain in the brain that represents a horizontal stimulus, but that domain may be subdivided. And across half of its region, there may be cells that prefer upward motion of that horizontal bar, and in the other half of that domain, we may have a clustering of neurons that prefer downward motion of that horizontal bar. This is what we call direction selectivity. And as Hubel and Wiesel discovered, there are numerous neurons throughout the visual pathways that respond best to motion presented in just one direction, with much weaker responses, if at all, in the opposite direction. So neurons in the visual cortex can be characterized by their orientation and directional selectivity. Visual cortical neurons can also be characterized by whether one eye or the other drives the activity to a greater or lesser degree. This is called ocular dominance. Now let's return to the organization of the visual pathways for just a moment to explain where this property arises. So as the two retinas send their axons into the optic tract, representing the same side of the visual world, it might be quite natural to assume that that information is combined in the lateral geniculate nucleus, but it's not. The lateral geniculate nucleus is a layered structure, and alternating layers of the lateral geniculate nucleus receive input from one retina or the other. Actually, there are two inner layers that are a bit different from the four outer layers. And the two inner layers are divided into these two components where one layer, layer 1, receives input from the contralateral eye, layer 2 from the ipsilateral eye, and then the four outer layers are divided with layers 4 and 6 receiving input from the contralateral eye, and layers 3 and 5 receiving input from the ipsilateral eye. So the point is that even within the thalamus, the information from the two eyes, while it's representing the same half of the visual world, it's still monocular at the single cell level. In fact, as the lateral geniculate nucleus projects to the visual cortex, the information remains monocular, which is to say that these monocular layers of the lateral geniculate nucleus project in an interdigitated fashion to nearby regions in the visual cortex, as if my fingers are representing the inputs driven by either the left eye or the right eye. And you can see that my fingers of my left and right hands are interdigitating as the thalamic input reaches layer 4 of the visual cortex. This establishes a set of columns that we can see both physiologically and anatomically, and we call them ocular dominance columns. So the input layer to the cortex, layer 4, remains monocular. It's only in the upper layers of cortex, as layer 4 projects to the circuitry above layer 4, layers 2 and 3, does information from the two eyes begin to get mixed for the first time. And that mixing of input from the two eyes is quite important because this makes possible the phenomenon of stereopsis. And stereopsis is that component of binocular vision that requires two eyes seeing slightly different views of the same object. And this is a perspective of depth that is possible for objects that are near, within about a meter or so, is where it works best. And beyond that distance or so, the lines of sight become increasingly parallel and therefore increasingly similar. But for objects that are within about arm's length, this dimension of depth is really quite rich, as it's elaborated by convergence of these two monocular channels in the upper layers of the visual cortex. So here's how this works. Imagine that we are fixating with our two eyes on a particular location in space, and all objects that are closer to us than that fixation point are going to be seen by what we call non-corresponding regions of the retinas. That is, there are points on the retina that don't project to the same point in the thalamus. So these slightly offset regions of the retina project their axons centrally, and the slight disparity in inputs that are arising onto the same cells in the upper layers of the visual cortex from these two disparate locations of the retinas give rise to signals that allow us to appreciate the fact that an object at location A is near in depth relative to the fixation point. A similar phenomenon would occur for background objects, where we might have a stimulation of non-corresponding points, but now on the opposite side of the fixation point. So locations indicated by this letter A would give rise to near disparities, and locations at a distance indicated by letter C would give rise to far disparities. Of course, there are other cues about depth other than stereopsis. We can convince ourselves easily of this fact. All one has to do is cover an eye, and the world does not all of a sudden seem to collapse from 3D to 2D. As we move our head around, we can see the movement of foreground objects relative to background objects, and that gives us important information about depth. But nevertheless, stereopsis can be a quite valuable tool as we navigate the world around us, and especially as we interact with objects within arm's length. One can demonstrate for yourself stereopsis by getting two pencils and holding them out at arm's length and then bringing them together. And you can see how well you can get those tips to touch. So I'm not so bad at this. But if you close one eye and make stereopsis go away, we'll see if this presents any further challenge. So I'm going to close my left eye and now try the same touching of the pencil tips. Whoa, I'm not so good anymore. Okay, that's a little better. Let's try the right eye now. So I'm going to close my right eye. So let's try now closing my right eye and seeing how well I do with my left. And I seem to be worse when I try to use my left eye. Okay, so the increased performance of using your two eyes together is not so difficult to demonstrate. So next we come to the topic of parallel pathways. So we've been talking about parallel pathways in different contexts, first in the somatic sensory system, now in the visual system. So it should be no surprise to you to discover that there are parallel pathways, even arising from the very same retina, as visual information is processed and then projected back into the brain. If one were to look at the ganglion cells that are present in the retina, we find actually a rather rich assortment of ganglion cells. They can be characterized in different ways by size, by morphology, and as investigators over the years have done so, we've adopted a nomenclature for different types of ganglion cells in different species, and here's the nomenclature that is used in the primate retina. There are small cells that we call P ganglion cells, much larger cells that we call M ganglion cells, and then an intermediate type of cell that's called the K ganglion cell. And the different morphologies that we see here are associated with the different physiological properties. More so, these different types of cells give rise to axonal projections that terminate in different parts of the lateral geniculate nucleus. There are projections from those small P cells that terminate among the small-celled layers of the lateral geniculate nucleus. These are called the parvocellular layers. Parvo means small, and we've illustrated them here in green. These are layers 3 through 6. Meanwhile, the larger M-type ganglion cells terminate in the first two layers of the lateral geniculate nucleus, illustrated here in blue, layers 1 and 2. So conveniently, the M cells project to the magnocellular layers, and the P cells project to the parvocellular layers. Now the K cells project to a zone in between the layers. So there's a region between each of these layers that we call the coniocellular layers. It's a region where there's a band of white matter or lamina of white matter between these cell-dense layers that have the numbers. So the K cells terminate in those spaces. These different neurons in the lateral geniculate nucleus send distinct patterns of projection on up to the visual cortex. The parvocellular layers of the lateral geniculate nucleus synapse into the bottom part of layer 4, a zone in the primate that we call layer 4C beta. The magnocellular layers from the anterior part of the lateral geniculate nucleus send their axons into layer 4C alpha, right on top of the input derived from those parvocellular neurons. The coniocellular cells of the lateral geniculate nucleus do something just a bit odd. They don't project to layer 4 at all. Rather, they send their axons up into layer 3, where they synapse in these zones that are rich in oxidative metabolism. And you may have heard about them if you're more familiar with visual cortical anatomy or physiology. They're called blobs, literally blobs of high enzymatic activity for oxidative metabolism. So this is an example of parallel processing. We have a small-celled pathway that's concerned especially with form and with color that runs through the parvocellular layers of the lateral geniculate nucleus and into the bottom of layer 4, 4C beta. There is a large-celled pathway that runs through the magnocellular layers of the lateral geniculate nucleus. It's not particularly concerned with high spatial detail or color, but rather it's concerned with broad patterns of motion. And this signal is terminating in layer 4C alpha, just above that parvocellular termination. And what about the coniocellular layers? Well, these are the more mysterious of the lateral geniculate nucleus layers. They've been not as well studied, but they do give rise to this very peculiar projection up into layer 3. And we think these coniocellular cells may have a particular role to play in color vision. But that remains an issue for ongoing investigation. Now you may be wondering what happens to these parallel pathways once we get beyond the primary visual cortex. Well, this remains a matter of active investigation. It was very attractive to assume that the parallel organization is highly conserved and rather quite rigid. I think we now know that that's probably not the case, that there is likely to be some mixing of information from one parallel pathway to another. Nevertheless, when we look beyond the primary visual cortex, it's possible to recognize two broad streams of processing that emanate out from the primary visual cortex through a set of areas that we call extrastriate visual cortex. So recall that stria of Gennari helps us to recognize a cortex that we call the striate visual cortex, area V1, and then beyond that we see these concentric zones of cortex, almost like concentric rings that surround our primary visual cortex. And it was attractive to call them areas V2, V3, V4, and so on, to emphasize our presumption that they represented progressively higher levels in some kind of visual hierarchy. Well, that too remains an object of ongoing investigation. Nevertheless, it is safe to conclude that beyond our primary visual cortex, there are rings of higher order visual areas. Now, what we're looking at here on the right is a cortical representation as if it were possible to peel off the cerebral hemisphere, flatten out the cortex, and then map out these visual areas. Well, in order to make that hemisphere flat, we have to make a cut along the cortex and the calcarine sulcus so that we can relax this three-dimensional shape into a two-dimensional sheet. And that's what we're looking at here. Now I want to draw your attention to a particular location which is found out here in the inferior border region between the lateral temporal cortex and lateral occipital cortex. It's where we find a couple of visual areas that we call area MT, or sometimes we call it the MT plus region. And this is an area that was named for the corresponding location in the macaque brain where a middle temporal visual area was discovered that is enriched in motion-sensitive cells. So this region, area MT, which is roughly in this position in the human brain, likewise seems to be especially enriched in motion-sensitive cells. And in fact, it's from the circuitry here in this area MT plus that we derive our notion of motion, as our chairman of neurobiology here at Duke University likes to say. So this is a higher order visual area that represents the kind of specialization that we encounter as we get further away from our primary visual cortex. From this area MT, these motion signals seem to inform a spatial analysis of the visual world that is happening in the parietal cortex. So we can think about this parietal area as addressing the question, where am I looking? Or where are the objects that I'm looking at that happen to be in motion? So this dorsal parietal stream, as we sometimes call it, is about mapping the world, achieving a spatial analysis of motion patterns. It's not particularly concerned with detail, with form, with color, with the recognition of faces or the identity of objects that we happen to be observing. Those kinds of functions happen along a ventral processing stream that emanates from the primary visual cortex through its near higher order areas and then out into the inferior temporal lobe. So this processing stream addresses the question, what am I looking at? So we have our where and our what pathways. Where going dorsally through the parietal lobe, and then what running ventrally through the temporal lobe. Now you can imagine with this functional dichotomy that people can have brain injuries in the parietal pathway or the temporal pathway and have deficits in one domain of visual function or another. And that is exactly what is seen. People can have injury that result from, let's say, stroke or tumor or trauma that affects this where pathway, and consequently there may be deficits in understanding spatial relations in the world around us, or perhaps very particular deficits in building a notion of motion. On the other hand, there may be strokes or other kinds of brain lesions affecting the what pathway in the inferior part of the temporal lobe. Those kinds of deficits can be quite particular, affecting our ability to answer this question, what am I looking at? Or in human relations, who am I looking at? There's a particular region of this posterior occipital temporal gyrus in an area that's sometimes called the fusiform gyrus, where when damaged leads to a deficit in the ability of people to recognize faces. It's called prosopagnosia, which means face blindness, or not knowing the identity of human faces. People with such injuries will have problems recognizing the faces of even loved ones, even their spouse or their near family members. So it's really quite a tragic situation for those individuals. And you can imagine how devastating that must be to their social relations. Now, thankfully, we acquire other cues besides the visual that assist in recognition, and these individuals get along reasonably well using these alternative cues. Nevertheless, they remain impaired in their ability to understand and recognize the visual features that we associate with the human face. Now, it's important to emphasize that these people, they're actually not blind. What they fail to do is take the structure of the visual image that is reflected off of the human face and interpret that in terms of the identity. Nearby regions of this ventral or inferior visual pathway likewise seem to represent other kinds of objects that we encounter in our world, perhaps inanimate objects. So one might be able to see, but not be able to recognize. Well, this concludes this tutorial and this tour of visual processing. I wish we could spend more time on this topic. I think as you've picked up today, this is an issue that has been of concern in my own academic life, and I'd love to share with you more of my research. Perhaps I'll have a chance to do so when we move on a little deeper into the course. So I'll leave you here for now and look forward to speaking with you next time. Hello again, and welcome to this tutorial on the central visual pathways. Today we're going to try to simplify some of the complexity of the way that vision is processed in the brain by considering an anatomical framework for understanding how signals get from the retina all the way up through the visual parts of the cerebral cortex. We're going to again confront circuits that are genetically instructed. However, as we'll see in a later session, these are the very same circuits that have become the gold standard for evaluating how circuitry and function can change in early life as a result of experience. And as I suggested in a different tutorial, vision is among the most wondrous of all of our senses that has inspired poets and philosophers and lyricists, and yes, even brain scientists, to wonder with their curiosity inflamed about how vision works in the brain. So I'd like to share a few insights with you in this series of tutorials, but I will readily recognize how much we have yet to understand about how these aspects of cognition actually work in the human brain. Hopefully we'll get there sometime in the course of your career, wherever that may lead you. Well, we have some learning objectives today. I'd like for you to be able to describe the distribution of axons from retinal ganglion cells to the major processing centers in the forebrain in the brainstem that process vision. I want you to be able to really focus in on the central visual pathways and the topographic representation of visual space. We call this visuotopy, and I want you to be able to characterize visuotopy in the primary visual cortex, also known as V1. And lastly, I want you to be able to discuss beyond V1 what happens to visual signals as they enter what we call extrastriate visual pathways. Let's begin by reminding you that the retina is an extension of the brain. It's diencephalic. It arises from the lateral aspects of the diencephalon in a formation called the optic vesicle. This optic vesicle grows out from the diencephalon and it interacts with other tissues that form the eye. And this optic cup, this optic vesicle, goes on to form the posterior layers of the inner surface of the eye, including the pigmented epithelium and the neural elements of the retina. Now let's turn to consider how the adult retina is attached to the rest of the brain via what was once the stalk of this optic vesicle. And that stalk is going to contain the axons of retinal ganglion cells that are distributed to a variety of targets in the diencephalon and in the midbrain. Here in this figure, we have an illustration of the distribution of those retinal ganglion cell axons, and we'll consider first their distribution to the diencephalon. And that will include a major projection to the lateral geniculate nucleus. Here it is sitting back in the posterior and lateral and somewhat ventral part of the thalamus. This is the principal target of retinal ganglion cells, and this is the part of the thalamus that will relay visual signals to the visual cortex, and that cortical recipient zone will be called the primary visual cortex. This is the pathway that's responsible for most aspects of what we know as vision. But there are other targets as well. In the diencephalon, we have an interesting nucleus of the hypothalamus that receives direct projections from the retina. This nucleus is called the suprachiasmatic nucleus, and it's quite an interesting brain structure indeed. It has within its neurons a molecular clock. So this nucleus is the master generator of circadian rhythms for the body, and those circadian rhythms are entrained to our typical day-night cycle, and that happens through visual inputs. Now, we might have assumed that these inputs are coming from collaterals of retinal ganglion cells that are otherwise projecting into the lateral geniculate nucleus. Fairly recently, we discovered that that's not the case. These inputs are actually coming from a special set of ganglion cells that have their own opsin protein, meaning that they can transduce photons of light directly, even without the outer layers of the retina. So this is a projection that's not particularly concerned with image formation, rather it's concerned with the ambient light in the environment, telling the difference between daytime and nighttime. Well, beyond the diencephalon, there are inputs from retinal ganglion cells to the midbrain, and this includes two structures, a region known as the pretectum, and then part of the tectum itself, the superior colliculus. So the superior colliculus we'll talk about in a later session when we talk about motor control, but this is involved in orienting the movements of the eyes and the head. And so imagine a bolt of lightning strikes somewhere off in the distance. We rather reflexively catch that flash of light and then turn our eyes and our head to turn and look in that direction. Well, that's a visual stimulus that triggered that motor adjustment. And that visual stimulus is conveyed to the superior colliculus via this pathway from the retina back to the tectum. Just anterior to the tectum, and in a bit of transitional region between the midbrain and the thalamus, is a structure called the pretectum. Actually, it's a set of nuclei called the pretectal nuclei, and these nuclei are involved in accommodation. And this involves regulating the constriction of the iris to change the diameter of the pupil, as well as governing the activity of the ciliary muscles that will either allow the lens to round up if it relieves tension on those zonual fibers, or if that muscle is more relaxed, then the tension on those fibers pulls the lens into a flatter shape. So this is a complex of the transition between diencephalon and midbrain that's involved in focusing images on the back of the retina. We'll have a separate tutorial to talk in more detail about the circuitry of the pupillary light reflex, since it's such an important component of the physical examination. Now I want us to turn our attention to the topic of visuotopy. So the visual world projects onto the retina in the following way. Because the lens is an optical device, the image is inverted and left-right reversed. So as you can see here in this illustration, this letter F that we may look at in the environment ends up being projected upside down and backwards, simply because of a function of the optical properties of the lens. So let's define some conventions that will be helpful in talking about the way visual space is mapped in the brain. So if we look straight ahead, there's some kind of a fixation point that's indicated here by this F, and the vertical line that defines the middle of that visual space is called the vertical meridian, and we can imagine a horizontal line that cuts right through that fixation point. That's called the horizontal meridian. So the vertical meridian is the one that we'll spend most of our time thinking about, because it establishes the basis for understanding how do the visual fields end up being represented on the opposite side of the brain. Now, if we look at what the two eyes see a large region of visual space that has considerable overlap between what the left and the right eye can each see individually. Well, in standard practice, if you are evaluating someone's vision, you will ask them to cover an eye and evaluate the visual space that's seen by each eye individually. And then that space is represented separately in the kinds of visual field charts that we see in the middle of this figure. So on the left-hand side, this is the left monocular visual field, or the amount of visual space that the left eye sees. And on the right, we can plot out the visual space that the right eye sees. Now notice that there is some kind of a cutout here in the middle, where on the inferior part of that visual space there seems to be a bit of a piece missing. Well, as my drawing might suggest, that's because the nose is blocking the view of the opposite lower region of visual space from the eye in question. And so now we have more of a complete picture of why we see the space that we do. So if I can clear these drawings for just a moment and return your focus to this convention of representing what each eye sees separately. Now notice that what we've done is we've taken this space that both eyes see, called the binocular visual field, and we've separated it out. All right? So both eyes are capable of seeing roughly this much visual space. But there's a region on the temporal or lateral extreme of the view of each eye that is monocular. That is, it's only seen by one eye. And that makes good sense, because what we're talking about is the far periphery of our visual world. And that far periphery is seen by the nasal region of the retina. So my right nasal toward the nose margin of the retina is the only part of my visual sensory apparatus that can see far right in my visual field. And likewise, the nasal, or the close to the nose part of the retina of my left eye, is the only part of my visual apparatus that can see to the far left. Okay, it's very important to consider what part of space can be seen by the two eyes together or one eye separately, because that will help you understand where the problem might be along the visual pathway in a patient that you suspect of having a visual field deficit. Now, on the lower part of this figure, what we have is an illustration of how this space projects on into the retina. So I would suggest that the bottom part of this figure is perhaps less important. Just recall this optical principle of inversion and left-right reversing the image. Now let's try to use some of these conventions and now understand how the visual world is projected onto both retinas, and then how that information from the retina is projected back to the brain. So as we can see here, the fixation point is seen by the central portion of each retina. This is a structure called the fovea. So the fovea sits back along the central part of the retina, which is actually just to the nasal side of the optic nerve head. So, consequently, if we follow the lines of projection back from this illustration, we see that objects that are out here to the right side of our fixation point are seen by the nasal portion of the right eye and the temporal portion of the left eye. Consequently, those portions of the two retinas need to send their information together in order to develop a coherent and congruous representation of that region of the visual world. That's where the optic chiasm comes in. Now notice what happens in the optic chiasm. There is a crossing of the projections derived from the nasal retina. In human beings, roughly about 55% or so of the axons in the optic nerve are derived from the nasal retina, and they cross in the optic chiasm and terminate on the contralateral side of the brain relative to their origin. The remaining 45% or so come from the temporal portion of the retina, and those axons remain on the same side of the nervous system. So by the time you are posterior to the optic chiasm in this visual pathway. These axons are now derived from the two eyes, but they are seeing the same part of the visual world. So in the illustration that I've tried to make for you here, if we consider the left optic tract here, the left optic tract is now representing the right side of visual space. Specifically, that region of right visual space that's seen by the right nasal retina and the left temporal retina. Conversely, the left side of the visual world would be seen by the nasal retina of the left eye and the temporal retina of the right eye. And those projections, with nasal being crossed, send signals back to the right optic chiasm, such that the right optic chiasm is now representing the left side of the visual world. So the optic chiasm really solves the challenge of having two eyes in the front part of our face. One might have thought that it would be easier just to have a single cyclopean eye, but we are bilaterally symmetrical creatures. So we have two visual organs, and we make great use of that fact in the construction of depth perception based on the slightly disparate views of the two eyes of near objects. But that's a story for a later session. For now, let me just emphasize to you that the optic chiasm sorts out the fact that the two eyes see portions of the same part of the visual hemifield. And with the principle of contralateral representation at stake, it's necessary to sort out those central projections to bring what the two eyes see of the same region of the visual world together in the appropriate hemisphere. Well, once the axons of the optic tract terminate in their appropriate diencephalic targets, with respect to the image-forming pathway anyway, these signals reach the lateral geniculate nucleus. So here now, with a lateral view of the brain, we see the lateral geniculate nucleus, which again, it's the visual thalamus, the part of the thalamus that's receiving input from the optic tract, and it's the part that gives rise to projections to the part of the cortex that we call the primary visual cortex. Well, that's quite a distance. That primary visual cortex is in the banks of the calcarine sulcus on the medial aspect of the occipital lobe, and in order for axons to grow from the lateral geniculate nucleus to the posterior medial temporal lobe, these axons have to grow around the lateral ventricle. So these axons swing first laterally, and then they run posterior in the white matter towards the occipital lobe. Now, it's possible to recognize essentially two parts to this so-called optic radiation. There are the axons that are emerging and sweeping in the more dorsal and posterior direction around the atrium of the lateral ventricle. These axons are destined to terminate on the posterior bank of the calcarine sulcus in the cuneus gyrus. Meanwhile, there are axons that are emerging and sweeping down into the temporal lobe and then around the lateral aspect of the temporal horn of the lateral ventricle. These axons perhaps have a slightly different challenge to try to get around the portion of the temporal lobe that otherwise is providing an impediment to the growth of these axons. But they do so, of course, and they make their path back to the lower bank of the calcarine sulcus, where they innervate cortex in the lingual gyrus. Now, this more temporal sweep of the optic radiation has a special name. We call this Meyer's loop. So when you hear the term Meyer's loop, think about this temporal portion of the optic radiation. Now let's look at the organization of these inputs along the banks of the calcarine sulcus. So, as I've just mentioned, the calcarine sulcus is formed by the folding of the lingual gyrus and the cuneus gyrus. So we have the cuneus, which is the wedge. Cuneus means wedge, on the upper bank of the calcarine sulcus. And then we have this tongue-like structure down below. So this is the lingual gyrus. Lingual means tongue. Now, notice how the color code is indicating the topography of the projections from the optic radiation into this cortex. Now the cuneus gyrus is representing the lower part of the visual field, and that is because the region of the lateral geniculate nucleus that was supplied by retinal fibers is coming from the upper part of the retina. The upper parts of the retina are viewing the lower portion of the visual world. So once we get into the thalamus and now into the occipital cortex, we have a sort of inversion of the world relative to the way that it's projected into the brain, such that the cuneus is representing the lower part of visual space. Now, that leaves it for then the lingual gyrus to represent the upper part of the visual field. And again, that's simply because the part of the lateral geniculate nucleus that grows the axons that form Meyer's loop is receiving input from the inferior parts of the retina that see the upper part of the visual world. If we look at the way the visual world projects onto the retina, we see that the central part of visual space is relatively limited, just a few degrees. In fact, what the fovea sees is about the width of your thumb at arm's length. So that's a bit of a rule of thumb about what the field of view is of the fovea. So that thumb's width amount of visual space is actually perhaps the inner region here of what the macula might see. And yet it's given over a very large representation in the visual cortex. So what the fovea is seeing is represented in the posterior pole of the hemisphere, and increasingly peripheral regions of the visual world are represented in increasing anterior regions of the visual cortex along the banks of the calcarine sulcus. So far, what I've been describing for you is how the visual world is represented in the primary visual cortex. Well, there is a considerable amount of the cerebral mantle devoted to the processing of vision. Perhaps as much as 25% or so in the human cerebral cortex is representing vision. So there is a series of visual cortical areas beyond the primary visual cortex that have now been mapped in the human brain. For reasons that we'll discuss when we look in more detail at the cellular structure of the visual cortex, the primary visual cortex is also known as the striate cortex. So these regions beyond the primary visual cortex are called extrastriate visual cortex. And we see a number of them here represented in blue and in the orange colors and in the purple and in the yellow. And what we discover is that beyond the banks of the calcarine sulcus, which is back in this region, we have a series of basically concentric rings of visual areas that emanate around the occipital lobe. And if it were possible to unfold and flatten out the cerebral cortex, as can be done computationally in a representation of the human brain obtained in a magnetic resonance imaging experiment, we see these concentric rings now cut open and laid out. And there's ongoing study trying to define exactly how many areas we have here, and that's still a matter of some discussion. But what we find is an increasingly more abstract representation of particular aspects of our visual world. One particular strategy that our visual system employs is the process of parallel pathways. And we see that elaborated in our extrastriate visual cortex. Broadly speaking, there are pathways that are mainly concerned with what we are looking at. And so the answer to the question, what we are looking at, is often provided in terms of the detailed shapes and patterns and texture and color of the objects and the faces that we see in the visual world. Well, that information is supplied in a ventral pathway that runs through the inferior aspect of the occipital lobe into the inferior temporal lobe. Well, in addition to what we're looking at, one fundamental question in vision is, where are we looking? And so the answer to the question where is essentially a spatial answer, and it has to do with the location of visual objects in the environment and perhaps where they happen to be moving about in that environment. And that sort of information is provided by a pathway that runs through an important structure in the brain called MT. MT stands for Middle Temporal Visual Area, which is where it was discovered in the non-human primate brain. In our brains, it seems to be near the junction of the temporal lobe and the occipital lobe. And from area MT, this sort of spatial and motion selective information tends to arise and travel up into the parietal lobe. So area MT, as we'll see in a different session on visual cortical processing, is enriched in cells that develop for us a notion of motion, as my chairman in the Department of Neurobiology here at Duke likes to say. So our notion of motion is derived by neural circuits that interact between our primary visual cortex and area MT, and from there the signals are further elaborated into a broader scheme of our visual environment that's elaborated in the parietal lobe. So remember, in the temporal lobe we have answers to the question, what am I looking at? And in the parietal lobe we have answers to the questions about where am I looking, or where are objects moving in the environment. Well, I hope this tutorial has helped lay the foundation for you in your understanding of the visual pathways and the concept of visuotopy in particular. This will be a very important topic as we consider the impact of lesions or disease that might afflict some component of the visual pathway. It will be important for you to understand what kind of visual field deficits might you expect to find in patients with damage to particular visual components or particular visual pathways. Well, we will have an entire tutorial on that topic, and I'll see you then. Hello again. Welcome to this tutorial on visual field deficits. We're going to be talking about the complexity of the visual pathways and how that complexity can be simplified if you understand the basic framework for the central visual projections, and that will be of great assistance to you as you examine patients that have visual field deficits. We're going to be basing our knowledge on genetically constructed circuits that are the foundation for visual function in the nervous system. And understanding these visual pathways and using that knowledge to properly localize lesions or disease in your patients will go a long way towards promoting healthy living in the treatment of disease. Well, my learning objectives for you today are to describe the distribution of the axons of retinal ganglion cells to the major processing centers in the forebrain and in the brainstem. I want you to be able to discuss the topographic representation of visual space in the primary visual cortex and its anatomical basis in the organization of the visual projections. Now, both of these objectives carry over from a previous session on the distribution of information from the retina to the brain. So if you've not yet viewed the tutorial on the central visual pathways, then now's the time to do that before proceeding on to this session. But if you've already done so, great. My final goal for you at the conclusion of this tutorial would be to characterize, in appropriate clinical terms, the visual field deficits that are associated with damage or disease along the central visual pathways. Well, just to be sure you recall what I'm talking about, the central visual pathways are the distribution of retinal ganglion cell axons from the retinas to the brain. And that distribution goes to both sides of the brain, with the nasal retina giving rise to axons that cross in the optic chiasm, the temporal retina giving rise to axons that remain on the ipsilateral side of the brain, running into the ipsilateral optic tract. Once axons grow posterior to the optic chiasm. They supply various targets in the diencephalon, such as the suprachiasmatic nucleus of the hypothalamus and the lateral geniculate nucleus, which is the major destination of visual information that's going to be used for the construction of visual percepts. In addition to those diencephalic targets, visual information is relayed to the midbrain, specifically to the region known as the pretectum, which is really just between the midbrain and the diencephalon, and the superior colliculus. Well, our focus today is going to be on the image-forming pathway. That is the pathway from the retina to the lateral geniculate nucleus, and then from the lateral geniculate nucleus back into the primary visual cortex and beyond, into extrastriate visual cortex. Here from the lateral view, again, we find our lateral geniculate nucleus, which gives rise to the optic radiations. The optic radiations sweep around the lateral ventricle. Those that run through the parietal white matter are representing the lower visual field, and those axons travel and terminate in the cuneus gyrus on the upper bank of the calcarine sulcus, whereas the portion of the optic radiation that emerges from the more ventral aspect of the geniculate sweeps around the temporal horn of the lateral ventricle through the white matter of the temporal lobe, terminating on the inferior bank of the calcarine sulcus in the lingual gyrus. These axons that are running through the white matter of the temporal lobe represent the upper portion of the contralateral visual field. So what we have then is an inversion of the representation of the world in the brain, such that the opposite side of the visual field is represented in one hemisphere, and the upper bank of the calcarine sulcus is representing the contralateral inferior quadrant of space, whereas the lower bank of the calcarine sulcus is representing the contralateral upper field. So with that background in mind, let's turn our attention now to the analysis of injury or disease that afflicts some local region along this visual pathway. And before we actually look at representations of such injuries, I want to get some terms out in front of you that will be useful as we describe these lesions. And these terms seem a little bit burdensome, but actually they're quite descriptive. And I think if you can understand the meaning of the terms, then their proper clinical use will quickly become second nature. Well, let's begin with the term anopsia. Anopsia refers to a large area of blindness in one eye. So hemianopsia means blindness in one hemifield, that is half field, that is seen just by one eye. Quadrantanopsia means blindness in one quadrant within a hemifield that's seen by an eye. Now, we modify these words with a term homonymous and a complementary term heteronymous. So homonymous means the same side. So this refers to the same visual hemifield that is seen in each eye. For example, one can have a lesion in the central visual pathway that produces blindness on the left side of the visual field for one eye, and the left side, or the same side, of the visual field for the other eye. That's an example of a homonymous hemianopsia. So if the regions of blindness are not on the same side of the midline for the views of each eye, then we call that a heteronymous hemianopsia. So heteronymous means different side. This refers to opposite visual hemifields being impacted in the field of view of each eye. And then finally, the word scotoma refers to a much smaller lesion, a small area of blindness somewhere within a visual field. The blind spot that we each bear in our visual hemispheres, due to the fact that the optic nerve head has no photoreceptors overlying it, is one example of a natural scotoma. But of course, scotomas can arise because of focal injury to the visual pathways or to the cortex itself. Now here's an important figure that I want you to spend some time studying. This figure relates a series of locations along the visual pathways that potentially could be the location of injury or disease to a representation of the visual fields of each eye. And the convention in this representation is to use black shading to indicate blindness. So let's walk through each of these locations and visual field deficits in turn. First of all, let's orient ourselves to the brain. We are looking down through the brain from above. So notice that we have the right eye on the right side of this illustration and the left eye on the left side of the illustration. That seems quite fundamental, but it will be critical in your studying and your learning to be certain that you know which side of the brain you're considering. So in this figure, right is right and left is left. Okay, so let's consider what happens if there were to be an injury in the right eye or the right optic nerve. Well, that should be pretty straightforward. The right eye would be blind. So this would be simply blindness in the right eye. Now, let's imagine that we had a lesion not in the optic nerve, but right in the middle of the optic chiasm. This can happen sometimes when there is a traumatic injury that induces a shearing force along the midline of the hemispheres, or perhaps with a tumor that's growing out of the pituitary gland, which attaches right below the optic chiasm. Well, a lesion of the optic chiasm would cut the axons of the nasal retina that cross in the midline. And that results in blindness in the peripheral parts of our visual world, specifically blindness in what the nasal retinas see. And the nasal retina of the left eye sees the lateral or the temporal part of the visual world. The nasal portion of the right eye sees its lateral or temporal portions of the visual world. So when we put the two visual fields together, we have preservation of central visual space, but blindness to the left and right sides of our visual fields. And that's an example of bitemporal hemianopsia, or specifically a heteronymous hemianopsia. So let's see how we use those terms. Hemianopsia means half visual field blindness for what an eye sees, and then the further modifiers talk about whether it's concordant for the field of view of each eye. And in this case, it's not. It's the left visual field that is lost for the view of the left eye, and the right visual field for the view of the right eye. That's a bilateral or bitemporal heteronymous hemianopsia. Now let's consider the remaining lesions here, and let's continue to work our way back. So lesion C is a lesion of the right optic tract. So at this location, we've sorted out the principle of contralateral representation, such that in the right optic tract, we have the axons from the two eyes that see the left side of the midline. So damage to the right optic tract produces blindness in the left side of the visual fields that are seen by each eye. So here's also a hemianopsia, but now it's a homonymous hemianopsia, specifically a left-sided homonymous hemianopsia. The left side of midline is lost for the field of view of each eye. The same would essentially be the case if we jump all the way back now and look at what happens with damage to the right occipital cortex. If we imagined that this was a widespread lesion in the right occipital cortex, we would likewise produce damage to the field of view that's seen on the left. Now notice there's a little bit of a carved region of visual preservation just around the central part of the visual field there on the left side of the midline. This is called macula sparing, and there are a couple of ideas as to why this comes about. One idea is simply the fact that we have such a large representation of the macular region in the visual cortex, a brain injury following, let's say, a stroke or a trauma, very well may damage a wide region of the visual cortex, but potentially there is preservation of some small region that very well may be representing the macula and what it sees, just because this is the part of the visual field that is given the most cortical circuitry. Well, should that be the case, then we might expect there to be some sparing of that region of the visual field. Well, perhaps there's another explanation that has to do with how the retina sorts out what's called the line of decussation, which means where exactly is the division between the temporal and the nasal retina. But that's a story more for the aficionados out there. So if we wanted to kick that into the discussion forum, that would be fine, but I won't take the time to talk about that story here. So let's complete our analysis of this figure by considering lesion D. Now, lesion D is taken to affect the axons of Meyer's loop. And as you recall, the axons that are growing out to contribute the inferior portion of the optic radiation have to loop around the temporal horn of the lateral ventricle. They do so by forming this very distinctive array of axons that sweep first inferior and laterally around the temporal horn of the lateral ventricle, and then posterior and inferior along the white matter of the inferior temporal lobe back into the occipital lobe. These axons terminate in the lingual gyrus, and they're representing the contralateral upper part of visual space. So if we had damage to these axons, then what we would see is loss of the representation of the contralateral superior quadrant of the visual world. So this would be an example of a left-sided homonymous quadrantenopsia. Do you see how we used all of those terms? So it's left of midline. It's a quadrant that's lost, so a quadrantenopsia, and it's homonymous. That is, it's the same side of the midline that's lost in the visual field of each eye. Okay, I'm going to leave you with a couple of study questions that will ask you to think about two patients who have problems that you suspect may be related to visual field deficits. So consider these patients and the options that I'm giving you to work through and see what you think. I'll see you next time. Welcome back. In this tutorial, I'd like to talk to you briefly about the circuitry that's responsible for the pupillary light reflex. Again, our journey into the visual system is an attempt to make a bit more simple some of the most complex pathways and function of the brain itself. We will once again consider circuitry that is genetically determined and provides a foundation for one very useful aspect of nervous system function, one that is exploited from a clinical point of view to assess the integrity of cranial nerves and brainstem circuits. That is the pupillary light reflex. So my learning objectives for you are to describe the distribution of retinal axons from ganglion cells to their major processing centers in the forebrain and the brainstem, and we have some particular brainstem targets in mind in this tutorial. And I want you to be able to use that knowledge to discuss the neuroanatomical basis for the pupillary light reflex. So let's consider again the distribution of the axons of our retinal ganglion cells. Those that reside on the temporal side of the retina grow their axons out through the optic nerve, and they remain on the ipsilateral side of the optic chiasm, being distributed to the appropriate targets in the diencephalon and in the midbrain. Those axons that arise from nasal retinal ganglion cells that sit here in this location, close to the nose on what we call the nasal side of the retina, they grow their axons through the optic nerve, and when they get into the optic chiasm, now these axons cross the midline. and they give rise to projections to a variety of targets in the diencephalon and in the midbrain, including the hypothalamus and the lateral geniculate nucleus of the diencephalon, and the superior colliculus and the pretectum in the midbrain. And for this discussion today, the projection of interest is this projection to the pretectum that I would highlight here, from the retina into a set of nuclei that sit in a transitional region between the diencephalon and the midbrain. And in that region of the dorsal part of the midbrain, we have the superior and inferior colliculi that collectively is called the tectum. Tectum means roof. It's the roof over the cerebral aqueduct. So the region just in front of the roof is the area of our focus in this session. That's what we call the pretectum. That's where we find a collection of nuclei that receive direct input from the retina. And it's those cells that will coordinate the distribution of that sensory information to the motor structures that result in the constriction of the pupils. So let's look now at a view just specifically of this reflex circuitry. So now what we see are the sensory and the motor limbs of this reflex arc. And let's first consider the sensory limb. Now, the retinas are projecting into the pretectum. And notice that the pretectum in one side of the midline is getting input derived from both retinas. So the temporal retina of one eye, the nasal retina of the other, are both contributing input into that pretectum. So that's important. So you need to understand that the sensory signals going into one side of that circuitry are derived from both eyes. Now, the motor limb of this reflex circuitry arises out of the pretectum. Now, notice another very important fact about this circuitry here that pertains to sidedness. The projections from these pretectal nuclei to the motor nuclei in question are bilateral. So here is our pretectal nucleus, and it gives rise to axons that are directed to both sides of the midline. And specifically, the target there is a small parasympathetic preganglionic nucleus called the Edinger-Westphal nucleus. It sits just dorsal and slightly lateral to our somatic motor nucleus, our ocular motor nucleus. And it's in this Edinger-Westphal nucleus that we have the preganglionic neurons that grow axons out through the third cranial nerve. So here in green is a Edinger-Westphal nucleus, and it's growing an axon out through the third nerve, which makes a synapse on a postganglionic neuron in the ciliary ganglion, and then that neuron grows its axons into the eye itself to supply ganglionic parasympathetic innervation of the constrictor muscle of the iris. So when this motor signal is conveyed down that third nerve, the iris will constrict. Now notice one other important fact that's worth emphasizing about the organization of the motor limb of this reflex. Once the signals leave the Edinger-Westphal nucleus, they remain on the same side. That is, they remain ipsilateral. So there is bilateral distribution of sensory signals into this circuitry, and then from the pretectum to the motor output, there is once again bilateral distribution of information. But once we get to the parasympathetic motor nucleus, the information is unilateral. Specifically, it remains ipsilateral. Okay, now I'm going to want you to think through systematically all the various perturbations of the pupillary light reflex. Imagine shining a light in one eye and looking for constriction of the pupils, shining a light in the opposite eye and doing the same. And imagine what you might see in a variety of patients. There will be hopefully all of your patients, but we know sadly that won't be the case. Hopefully all of your patients would show symmetrical bilateral constriction of the pupils. And this bilateral constriction is an indication that the signals are being processed in the pretectum distributed bilaterally to the motor nuclei, and these signals are being sent along the efferent pathway to the constrictor muscles of the iris, leading to the symmetrical constriction of the pupils. Now, if any of that is not observed, then you very well may have an injury to the eye, to the optic nerves, to the optic tract, to the circuitry of the pretectum, to the region of the Edinger-Westphal nucleus and the nerve roots of the third nerve, or the path taken by those preganglionic or postganglionic axons out to the constrictor muscles of the iris. And it's your job as a clinician to reason through this circuitry and make a judgment as to whether you think there is a localized lesion. Now, our focus here is on the constriction of the pupil, but let me just say in passing that there's also a dilation of the pupil that's under the governance of the sympathetic nervous system. And as you may recall, the sympathetic outflow comes from preganglionic neurons in the intermediolateral cell column of the thoracic spinal cord. So there's a rather circuitous route that the neural signals take to get out of the brain into that sympathetic chain of ganglia and then back to the iris. So just keep in mind for our consideration here that pupillary constriction is under the governance of the parasympathetic outflow through the third nerve, whereas pupil dilation is under the governance of the sympathetic nervous system. So I'd like to leave you, after this brief tutorial, with 3 study questions. And these are questions that will present clinical vignettes from 3 patients, each of whom has some sign associated with your performance of the pupillary light reflex. And your job is to consider whether there might be a localized lesion to either the sensory limb of this reflex, the motor limb, or perhaps the central stations in the pretectum where sensation is integrated to produce motor action.